Hey everybody, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, and as you probably know by now, I'm coming to you from suburban Philadelphia, where I teach anatomy and physiology at Bucks County Community College, and I've been doing that since 2002. I have some exciting news for you. Recently, I've begun sharing some tutor video clips to my Instagram account. These clips come from Anatomy and Physiology Digital Suite, which is my McGraw-Hill project that's kind of like a textbook, but instead of reading a book, students watch 5 to 6 minute tutor videos I've made that cover most of the concepts in A&P. So if you haven't yet, go to my Instagram account, StudentHelp4AP, that's Student Help, the number 4, AP, and follow me and see the clip of the day. You can also go to my YouTube channel, StudentHelp4AP, and watch my older 2-minute tutor videos to help you understand some of the key concepts in A&P. If you like any of these things, please tell your classmates, your friends, and your professors. Help spread the word, and the more people I get using these and benefiting from them, the better. And if it helps you get your B or better in A&P, well, then that just makes my day. Today we're going to get into the lymphatic system. Hey, just a little side note here. I'm going to be talking about some lymphatic vessel anatomy during this episode, so I am posting an illustration of lymphatic vessels to my Instagram account. So if you can, as long as you're not driving, go to Instagram, look me up, Student Help 4AP, and find that image. And you can hopefully follow along a little bit with that image while you're listening. Okay, back to the show. We finished up the circulatory system, but there are other series of vessels in our bodies called lymphatic vessels, and they are also carrying fluids to other regions of our bodies. So I want to talk about the lymphatic system today. I want to get into the introduction to the lymphatic system. I don't think we're going to get through the whole thing in one episode, but this is a good one because the lymphatic system is very important clinically to a lot of healthcare practitioners. You hear a lot about lymph nodes, you hear a lot about lymphatic vessels, you hear a lot about the spleen and the tonsils, and those are all parts of the lymphatic system. But first, I got a question from a listener this week that I found really interesting. So I would like to address that first, and then we'll jump back into the lymphatic system. All right, I recently got a question from a listener. His name is Isaac. He didn't mention where he's from, so I don't have that information, but he asked me about VO2 max. What is it, and how do we test it? That is a great question, especially for someone like me who studied exercise physiology. So let's start by discussing oxygen and ATP, adenosine triphosphate. That is our energy currency. Since energy cannot be created nor destroyed, we don't produce energy. Rather, the mitochondria in our cells convert the energy that already exists in molecules like glucose to ATP via aerobic respiration or anaerobic fermentation. Aerobic respiration utilizes oxygen and yields far more ATP from one molecule of glucose. This is what we use during aerobic exercise like running, swimming, biking, tennis, etc. Now, VO2 max, or maximum oxygen uptake, is a measure of the maximum amount of oxygen your body can use during intense exercise. It reflects the efficiency and capacity of your cardiovascular and respiratory systems to supply oxygen to your muscles, as well as your muscles' ability to use that oxygen for ATP production. VO2 max is commonly used as an indicator of aerobic fitness and endurance. So how are we going to measure VO2 max? Well, there are two primary methods. There's the direct measurement and there's an estimation. The direct measurement you're going to do in a lab. This is the most accurate method for determining VO2 max and typically involves an exercise test in a laboratory setting. So what you're going to do is you're going to perform progressively more intense exercise, usually on a treadmill or a stationary bike or something like that. And the intensity will increase in stages, either by increasing the speed with which you are moving or increasing the resistance. While you're exercising, you're going to wear a face mask or some kind of mouthpiece connected to a machine that measures the volume of oxygen that you breathe and the volume of carbon dioxide that you exhale. So you're inhaling oxygen and you're exhaling carbon dioxide. This is done in real time, and the machine calculates the ratio of oxygen consumption to carbon dioxide production. That is your respiratory exchange ratio, or RER. When you produce ATP using oxygen, one of the byproducts is carbon dioxide. So by measuring how much oxygen you bring in and how much carbon dioxide you produce, we can see the efficiency with which you're using the oxygen. The test is going to continue until you can no longer maintain the required intensity. So once you become exhausted, the VO2 max value is usually recorded just before exhaustion, because remember, we're thinking about max. That's when the oxygen consumption levels off and does not increase with any increased effort. This is the point at which maximal oxygen uptake has been reached. Now, the lab can do a good job of getting a real precise measurement. You can also get an estimation in the field. So if you don't have access to a lab, there's a few field tests you can do to provide a good estimate of VO2 max based on exercise performance. They're not as precise than the direct measurement, but they can still give you some useful information. Some of these common tests include the Cooper test, which involves running as far as possible in 12 minutes. The distance covered is used to estimate VO2 max. There's also the Rockport walking test. This test involves walking 1 mile as fast as possible and then measuring the heart rate immediately afterward. Based on the time it took to complete the mile and the heart rate response, you can get an estimation of VO2 max. And then there's also the Bruce treadmill test. This involves walking on a treadmill at increasing speeds and inclines, and VO2 max can be estimated based on the time to exhaustion. So how long does it take you to go to complete failure? If we're increasing how fast you're walking and the resistance, which would be the incline. VO2 max is basically one of the best indicators of aerobic fitness. A higher VO2 max indicates better cardiovascular health and a greater capacity for endurance exercise. And there's a lot of things that can influence our VO2 max. Obviously, genetics is always going to be there. Some people just naturally have a higher VO2 max due to their genetic makeup than other people do. You can also train to have a higher VO2 max. Aerobic training like running, cycling, swimming, tennis, other aerobic sports like I mentioned earlier, they can improve your VO2 max. You can train to have a higher VO2 max. Age and gender. VO2 max typically declines as we get older, and biological males tend to have higher VO2 max than biological females on average. This is due to the difference in muscle mass that you typically see and also cardiovascular function. When we're measuring VO2 max, we typically use millimeters of oxygen consumed per minute per kilogram of body weight. So milliliters per kilogram per minute. On average, you would see a sedentary adult have about 25 to 40 milliliters per kilogram per minute. More active individuals, you would see 40 to 50. And the elite athletes, we typically see somewhere around 60 to 80 milliliters per kilogram per minute. Now, some of the things that can decrease your VO2 max are body composition. People who have a lower lean body mass, which means a higher body fat percentage, that will lower your VO2 max. Being in high altitude. Increased altitude lowers VO2 max. And smoking, obviously. Smoking cigarettes does a lot of damage to your lungs, which will obviously change the way you process oxygen. So I hope that answers questions about VO2 max. This is a really interesting topic for me because when you talk about human performance, when you talk about athletes, VO2 max is one of the number one things we're looking for to increase performance, especially athletically. And I'm not just talking about people who play sports. This is important for anyone who wants to be in charge of their own cardiovascular and respiratory fitness and overall health. And you might have an occupation that requires you to be very physically active and have a lot of endurance. And what about dancers, right? Dancers have to be just as athletic as cyclists and marathon runners. So this is an important aspect of just about anyone's own personal health. So once again, Isaac, thank you so much for sending in that question. I really hope that I've been able to answer it effectively for you. And anyone else who's interested in VO2 max, I hope that you benefited from that. Okay, so remember when we talked about capillary exchange in the circulatory system, we discussed the fact that blood plasma filters out of the capillaries into the interstitial fluid, and interstitial fluid gets reabsorbed back into the blood capillaries, and then it moves on as blood plasma. Well, about 15% of that interstitial fluid does not get reabsorbed into the bloodstream, and instead, it makes its way into another series of vessels called the lymphatic system. When this happens, the fluid is called lymph, and lymph has a very specific role. So in addition to those blood capillaries, we also have lymphatic capillaries, and they're microscopic vessels, and they absorb that extra interstitial fluid. There's a hydrostatic pressure gradient that exists, and it results in the flow of interstitial fluid into the lymphatic capillaries, where it then becomes that lymph. The combination of lymph, the vessels that carry it, the cells that make up lymphatic tissue, and the organs that process lymph is called the lymphatic system. Now, we also have lymphatic vessels and lymphatic trunks leading back to the blood vessels. So eventually, that lymph is going to get back into blood plasma. It is temporarily in a different location and providing a different function. But we have to return that fluid back into the circulatory system. And there are some organs that are involved in the process of that lymph. They are the spleen, the thymus, the tonsils, the red bone marrow, and about 450 small lymph nodes that you can find all over your body. Now, in general, lymph is a clear liquid that is similar to blood plasma, but it has lower protein concentrations. And what's interesting is that the composition of lymph is going to vary from location to location in your body because different tissues have different functions. It also depends on the body's activities and the current state of illness or infection that might exist in your body. For this reason, lymph may consist not only of water, but of viruses, bacteria, dead cells, proteins, lipids, white blood cells, hormones, and even cancer cells that have been freed from their primary location and could travel to another location in the body. Those cancer cells are called metastatic cancer cells, which is one mechanism of how cancer spreads from one tissue to another. Cancer can also spread just by touching another tissue. We call that direct extension, or it can actually spread through the circulatory system. But you may have heard of when someone has cancer, they check the lymph nodes to see if there are any cancer cells in the lymph nodes because that is one of the indicators of whether or not those cancer cells are spreading to other organs and tissues. Now, the lymphatic system has three main functions. The first one is to recover the interstitial fluid that didn't get reabsorbed back into the bloodstream, right? We talked about that. About 3 liters of water and up to half of the blood's plasma proteins would accumulate in the tissues every day if the lymphatic vessels didn't deliver it back to the circulatory system. And if someone doesn't have proper lymphatic drainage, you see that because their tissues get really swollen. We call that lymphedema. The lymphatic system also participates in immunity because it picks up foreign cells, cellular debris, and cellular chemicals and takes them away from the tissues. The lymphatic organs that receive the lymph are loaded with immune cells like lymphocytes that will fight foreign cells and initiate immune responses in the body to defend against disease. You may have noticed that when you're sick, even if you just have a cold, you might feel under your jaw, you'll feel some swollen lymph nodes. That is because those lymph nodes are getting inflamed with all that extra lymph coming from the infected tissues, as well as all of the extra white blood cells that you've been producing, all of the viruses or pathogens that are infecting you. They're all going to make their way through the lymphatic system into those lymph nodes where they can be processed. In the small intestine of the digestive tract, lipids we consume are absorbed by the blood capillaries, but also by lymphatic vessels. We call those vessels lacteals, and they absorb dietary lipids, fats that we consume, so they can be delivered to the bloodstream further down the line, like later on, not immediately or directly. Now, we not only have lymphatic organs, but we have lymphatic tissue found in the mucous membranes of several organ systems, like the respiratory tract, the digestive tract, the reproductive tract, the urinary tract. The difference is that in lymphatic organs, like lymph nodes, for example, the tissue is made of densely packed cells. But in the mucous membranes, the cells are more loosely arranged and scattered. There are several different cells in lymphatic tissue, and many of them are derived from leukocytes, or white blood cells. And each cell has specific lymphatic functions. If you review episode 40 on leukocytes, or white blood cells, you can hear about a lot of those functions. Let's talk about lymphocytes, though. Remember, lymphocytes are a kind of white blood cell. And we're going to talk about 3 types of lymphocytes: B lymphocytes, T lymphocytes, and natural killer cells. B lymphocytes, they initiate the production of antibodies to defend against specific pathogens we become exposed to. So think about having chickenpox, or having the measles, or even just a regular common cold or flu. When you have those infections, your body is going to react to that by producing proteins called antibodies. And these are produced by cells that come from B lymphocytes. Those antibodies are designed to attack or bind to the part of the pathogen called an antigen. And by doing that, it can render that pathogen harmless against you. Now, you still get sick the first time because it takes some time to generate those antibodies. But then the next time you become exposed to that pathogen, you don't get sick because you have antibodies against the pathogen that will go and get them and render them harmless against you. T lymphocytes, which mature in the thymus, which is a gland found superior to your heart. It is typically pretty shrunk in adults. It's much more active in fetuses and very young children. That's the thymus. T lymphocytes mature in the thymus, which is why they're called T lymphocytes, and they directly attack foreign cells or our own cells that have been infected by a pathogen. There are many types of T lymphocytes involved in immunity, and some we'll talk about later on in another episode when we get to the immune system.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times. But we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
Natural killer cells have a similar function to T lymphocytes. In addition to attacking bacteria and virus-infected cells, they also destroy cancerous cells and cells that are transplanted from a donor. This is why when someone receives an organ transplant, they are given immunosuppressive drugs that will decrease the immune system because you don't want your body rejecting that organ or tissue. And the immune system will do that because it will recognize that those cells lack your self-antigens. And those white blood cells are good at recognizing what is not you and what shouldn't be there. Now, another kind of white blood cell, neutrophils, not lymphocytes, defend us against harmful bacteria in the body. So that's another type of cell you'll see inside lymphatic organs because they're good for going after bacteria. So there are cells that are going to look at these foreign cells that come into our body and present the antigens of those cells to T lymphocytes so they can be aware of this foreign invader. And we call those cells antigen presenting cells, or APCs. Let's talk about 3 different kinds of antigen presenting cells. One is macrophages. Macrophages are phagocytes that are derived from monocytes, a kind of white blood cell. So they phagocytize bacteria, which means they ingest it, they pull it into the cell, destroy it, and then spit out all of the debris. And they do that with bacteria, with foreign matter, with tissue debris, and dead neutrophils. In episodes 9 and 10, I discussed the integumentary system, and you can go back and review the epidermis, and you'll remember that there are 5 different epidermal cells. Melanocytes was one. They produce melanin. We had basal stem cells. We have keratinocytes. We had tactile cells. And we also had dendritic cells, which are part of the immune system. Well, they're not only found in the epidermis. We also see them in lymphatic organs and mucous membranes. They utilize receptor-mediated endocytosis to engulf foreign matter before they migrate to a lymph node so an immune response can be initiated. And then finally, we have reticular cells. They don't migrate, but they remain in the stroma of lymphatic organs, like lymph nodes and the spleen, where they can process foreign matter after it's made its way to the lymphatic organs. Okay, so the cells are one thing, but also we've got these vessels that have to carry the lymph throughout our body. We see lymphatic vessels in nearly every tissue of the body. So the interstitial fluid that doesn't get reabsorbed by the circulatory system can be processed, and we can get rid of all the foreign matter, dead cells, debris, pathogens, etc., all because of those vessels. We call these lymphatic vessels lymphatics. That's just a short term for it that a lot of people use. And they carry lymph from the tissues to the lymphatic organs and then deliver it to the blood plasma. So let's consider the path that lymph has to follow, beginning with where the capillary exchange is taking place in the tissues. So lymphatic capillaries are right next to blood capillaries. And sometimes we call them terminal lymphatics because they're not connected to any other vessels. They just start there with the blunt end absorbing interstitial fluid. The only tissues that do not have lymphatic capillaries are bone, bone marrow, cartilage, and the outer surface of the eye, which is the cornea. The endothelial cells of the lymphatic capillaries have no basal lamina, and they're not joined by tight junctions like we see in blood capillaries. Instead, they slightly overlap each other, leaving some gaps between them. And the interstitial fluid, its solutes, bacteria, lymphocytes, and some pretty large particles can actually flow into the lymphatics and become lymph. The lymphatic capillaries are held tight to their surrounding tissues by anchoring filaments, which are proteins that hold the outer flaps of those endothelial cells to the surrounding cells, keeping those large gaps open. Without those large gaps, we can't fit these pretty big molecules and pathogens and solutes and put them into lymphatic vessels. So using those proteins to hold those gaps open really helps get the interstitial fluid into the lymphatic capillary to become lymph. This is all pressure dependent. When the hydrostatic pressure in the lymphatic capillary is higher than in the tissue fluid, the inner flaps are actually pushed against the outer flaps, and those gaps are closed. And then interstitial fluid can't get in. So when the lymphatic capillary is getting too filled with lymph, that's going to create a pressure gradient that closes those gaps. But due to filtration of blood plasma, if the hydrostatic pressure of the interstitial fluid increases, that will push open those gaps by pushing the inner flaps inward. And those gaps get bigger, they open, and the tissue fluid can make its way into the lymphatic capillary with its solutes and become lymph. Once we get out of the capillaries, we get into larger lymphatic vessels, and they're more similar to veins because they have like a similar embryological origin. Like veins, they have 3 layers or tunics, and they also have one-way valves that prevent the backflow of lymph. So if you remember from the circulatory system, a lot of our veins have valves to make sure blood can't fall backward further from the heart when it's really supposed to be getting closer to the heart. Some of the larger lymphatic vessels have valves like this as well. The lymphatic system also uses skeletal muscle contractions and the respiratory pump to drive lymphatic flow against gravity. So when you're walking and your skeletal muscles are contracting, they're squeezing the lymphatic vessels. And that is pushing the lymph further along in the vessel. And then the valves keep it from falling backward when you relax those muscles. And then in your abdominal and thoracic cavities, when you breathe, you create pressure gradients that do the same thing. Also, like veins, the vessels get larger and larger each time they converge, taking lymph to its destination. Lymphatic capillaries lead to vessels called collecting vessels. The collecting vessels converge into 11 different lymphatic trunks. Those lymphatic trunks deliver lymph to 2 collecting ducts. The collecting ducts deliver the lymph into the blood plasma by way of the bilateral subclavian veins. So the collecting ducts of the lymphatic system deliver their contents directly into the subclavian veins. So we're returning the lymph into the blood plasma. Now, there are specific names for these vessels, and they're typically named for their location. We have the intestinal trunk. There's only one of those. We have lumbar, intercostal, and bronchomediastinal trunks. They're bilateral. We also have subclavian and jugular trunks that are bilateral. So we have a total of 11. The intestinal trunk gets its lymph from most of the structures found around the abdominal region, so in the abdominal cavity area. The lumbar trunks get their lymph from the lower limbs, the abdominopelvic wall, and the pelvic organs. The bronchomediastinal trunks get theirs from the deep structures of the thoracic cavity. The subclavian trunks get them from the upper limbs, the breasts, and the superficial thoracic wall, and the jugular trunks, as you can imagine, get their lymph from the head and neck. Now there are 2 lymphatic ducts, and they are called the thoracic duct and the right lymphatic duct. Now let's think about the anatomy a little bit. So I'm going to try to describe this, and I know you don't have an image of it, but maybe I'll be able to post an image on my Instagram account for you to take a look at. The intestinal and lumbar trunks receive lymph from the digestive system and merge to form a structure called the cisterna chyli, which gives way to the thoracic duct. As the thoracic duct ascends the body through the diaphragm and into the thoracic cavity, lymph is delivered to it from the left bronchomediastinal, subclavian, and jugular trunks. After that, the thoracic duct empties its lymph into the left subclavian vein, where it is returned to the circulatory system and becomes blood plasma. The right lymphatic duct receives lymph from the right bronchomediastinal, jugular, and subclavian trunks and delivers its lymph into the right subclavian vein. All right, I think that's good for this episode because I don't want to get started into the lymphatic organs yet because that's going to be a lot. So I think we'll dedicate the next episode entirely to lymphatic organs, where we'll talk about the spleen and the tonsils and lymphatic tissues and things like that. And that'll take up a good episode right there. Once again, thank you to Isaac for sending me the question about VO2 max. I had a lot of fun discussing VO2 max. Again, exercise physiology person here. That was really interesting for me, and I hope that you enjoyed that discussion as well. Don't forget, check out my Instagram account, Student Help for AP. And check out all the video clips that I'm posting on Instagram right now. And check out the ones that I already have on my YouTube channel. And hopefully they can really help you get that B or better that you need in A and P to move on. Thanks again for listening. I really appreciate it. Have a great day, and good luck on your next exam. I'll see you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media. Don't forget to check out my Instagram account and my YouTube channel, both called Student Help for AP. I'm posting a lot of stuff on there right now that you can watch and that will hopefully really help you understand this A&P stuff. Very special thank you out to my family, Bucks County Community College, and McGraw-Hill Education, where you can find my tutor video-based textbook alternative Anatomy and Physiology digital suite, already being used by several colleges and universities throughout the United States.